Sir, welcome to PK's classes. Today we are going to study enzyme linked immunosorbent assay or ELISA. ELISA. So enzyme linked immunosorbent assay, in short form it is called ELISA, or we can say enzyme immunoassay, EIA. It is a plate-based assay technique, and we call it as immunoassay because there will be interaction between antigen and antibody. We can also call it as non-isotopic assay. Non-isotopic because there is another assay technique called RIA, radio immunoassay, where we use the radioisotope, and here we are not using this radioisotope, so it is not as hazardous as radio immunoassay because there is no radioisotopes involved. So we can also call it as non-isotopic immunoassay. So let us discuss the principle of this ELISA. Basically, it is an immunoassay where enzyme is linked. So the difference between radio immunoassay and ELISA is, here a radioisotope is bound instead of enzyme in case of RIA. In ELISA, enzyme is linked. So first, immunoassay. So when we say immunoassay, that means there is immune reaction. That means there is reaction between antigen and antibody. So this assay we use to either detect the antigen or the antibody in the sample, say blood, say urine. So we can determine the presence of antigen or antibody, and we can also quantify how much antigen is present or how much antibody is present. Suppose the sample contains antigen, then we can use one antibody specific to that antigen. Suppose the sample contains antibody, then we can use antigen specific to that antibody. And in this, what we do, we link the enzyme with the antibody, and that enzyme has a substrate. So when we add that substrate to the mixture of antigen and antibody, and that antibody is conjugated with enzyme, then the substrate will be converted to a colored product. So there will be change in color. So if there is change in color, that means the antibody conjugated with the enzyme is binding with the antigen. That means the antigen is present. Or if the sample contains the antibody, then that antibody will react with the antigen, and then we will use, so this will be the primary antibody, first degree or primary antibody. Then we will combine the secondary antibody, that is conjugated with the enzyme, and then this enzyme will convert the substrate to a product which is a colored product. So the antibody will be conjugated with the enzyme, and that can directly bind with the antigen, or can indirectly through the sample antibody it will bind with the antigen, and if it binds, then it will be present in the mixture. So when we add the substrate, that substrate will be converted to a product by the enzyme, and that product is a colored product. From the color we can say whether the antigen is present or the antibody is present, and from the intensity of the color we can also say how much antigen is present or how much antibody is present. This is the principle. So if the sample has the antigen, then we will use one antibody conjugated with the enzyme. If the sample is the antibody, then we will first we will allow it to bind with the antigen. Then we will add another antibody which has affinity for this antibody in the sample, which is conjugated with the enzyme. We call it as the secondary antibody, and that enzyme converts the substrate into a colored product. So this is the basic principle of ELISA. So you can see two types of enzyme you usually use. One is horseradish peroxidase and the other is alkaline phosphatase. So in the horseradish peroxidase enzyme, the usual substrate is H2O2 and the product obtained is blue in color. We can also use OPD, O-phenylene diamine dihydrochloride, where the color of the product is amber color. We can also use TMB 3,5,5-tetramethyl benzidine. The color of the product obtained is blue. We can also use ABTS 2,2-azino bis 3-ethyl benzothiazolene 6-sulfonic acid, and the color of the product obtained is green. So if we use the enzyme horseradish peroxidase or HRP, then we can use all these four substrates, but the most commonly used substrate is hydrogen peroxide, and the colored product has blue color. So this is commonly used. And if we use the enzyme alkaline phosphatase, then the substrate used is paranitrophenyl phosphate, that is PNPP, and color produced here is yellow color. So these are the two most commonly used enzymes, and they have specific substrates, and then the color produced is also different. Now let us see, there are different types of ELISA. There are basically four types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So what happens in direct ELISA, why we call it as direct ELISA? In this case, there is only one antibody, and that antibody reacts with the antigen, and that antibody is conjugated with the enzyme which can convert the substrate into a colored product. So you can see here the enzyme conjugated antibody directly binds with the antigen present in the sample. So this is called direct ELISA. If the antigen is not in direct contact with the enzyme conjugated antibody, it is connected through another antibody, then it is called indirect. So you can see here antigen binds with an unconjugated antibody, that is not conjugated with any enzyme. So antigen binds with one antibody, we can call it as primary antibody, first degree primary antibody. Then this primary antibody binds with another antibody conjugated with the enzyme. So we can call it as the secondary antibody. So primary, the first one, and to the primary antibody, the secondary antibody binds. So this secondary antibody is conjugated with the enzyme. So the enzyme conjugated antibody is not in direct contact with the antigen, so we call it as indirect. Okay, so in this indirect ELISA we can detect either antigen or antibody. Mostly we use it for antibody. Here this primary antibody is the antibody present in the sample. If we want to detect the antigen, then we have to use one primary unconjugated antibody followed by the enzyme conjugated secondary antibody. So for direct ELISA, we can detect antigen only. In indirect ELISA, we mostly detect antibody. However, we can also detect antigen. Then the third one, sandwich ELISA. We call it as sandwich. You must have ate this sandwich. Sandwich, here the sandwich means antigen is sandwiched between two antibodies. One antibody below and one antibody above, and between these two antibodies there is the antigen sandwiched. So we call it as sandwich ELISA. And here in this sandwich ELISA, we have this antibody. First, that antibody we can call it as capture antibody, which will be bound to the plate, and then we'll add this antibody conjugated with enzyme. So if we use directly only one antibody in addition to the capture antibody, then we can call it as direct sandwich. And if we use two antibodies, one primary unconjugated antibody, then secondary enzyme conjugated antibody, then we can call it as sandwich indirect ELISA. So basically it is called sandwich ELISA because the antigen to be detected is sandwiched between the capture antibody and the detection antibody. So the detection antibody can be conjugated with the enzyme, or the detection antibody is unconjugated, then we have to add another antibody conjugated with the enzyme called the secondary antibody conjugated with the enzyme. So that is called sandwich. So in the sandwich ELISA, we can detect again antigen. Okay. So this is the difference. And the fourth type of ELISA is the competitive ELISA. That means when we cannot sandwich the antigen then between two antibodies, then we use this competitive ELISA. That means suppose there is one antigen, and to this antigen we will add two types of antibodies, one unconjugated and the other conjugated with the enzyme. Okay, so unconjugated means suppose this is from sample, so the one antibody from the sample and the other enzyme conjugated antibody. So there will be competition between the antibody in the sample and the enzyme conjugated antibody. If the antibody in the sample is more, then that will bind to the antigen, and if the antibody in the sample is less, then this enzyme conjugated antibody will bind to the antigen. So that is competitive ELISA. So we can discuss them in later classes in detail. So basically in competitive ELISA, we see that the antibody present in the sample, if it will be more, then it will bind to the antigen, and if it will be less, then it will not bind to the antigen. And if we have added another antibody which is conjugated to enzyme, when the antibody in the sample will be less, this enzyme conjugated antibody will bind. So there will, when we add the substrate, that substrate can change to a colored product. So substrate is changed to a colored product in presence of the enzyme. That means in this case, if there is change in color, that means the sample does not contain the antibody. That means test will be negative. And if here the color is not changing, that means antibody has the antibody in the sample is more, which is binding to the antigen. So in the competition between these two, who will win? Who is more in concentration? If the antibody in the sample is more, it will bind. If the antibody in the sample is less, then the antibody conjugated to the enzyme will bind. And when the antibody conjugated to the enzyme will bind, then only the sample, the substrate will be converted to a colored product. So accordingly, these 4 types of ELISA, and in later classes we will study in detail about these 4. So the basic process is we have to take either antigen or antibody, and then we have to add the sample, and we have to allow the antigen antibody reaction, and after each step we have to wash. We have to wash with phosphate buffer saline or PBS, which provides the same environmental condition as blood or serum. So it provides the same pH because its pH is 7.4. PBS, phosphate buffer saline, has pH 7.4. It also provides the same ionic concentration. So during washing we use PBS buffer. Then we use here a micro titer plate with wells. Usually 96 well micro titer plate is used. It can have more than that also, even 384 well micro titer plates are there. So the antigen or antibody can directly bind to this type plate because this plate is made up of plastic material, polystyrene or polyvinyl chloride, polycarbonate, cycloolefin. So these are the materials which are used to prepare this micro titer plate. And this micro titer plate has wells in it, and usually 96 well plates are used. So in this way wells are there, and we use these wells for this antigen antibody reaction. So this antigen and antibody, they can directly bind to this plate because the material is such so that the antigen and antibody can directly bind to it. So to avoid that, we have to also use surface blocking agents. They should be non-reactive proteins. They should not react with any antigen or antibody. They can only react with the micro titer plate. So that will prevent binding of antigen or antibody with the assay plate. So suppose we are now coating this plate with antigen, this sample antigen. So once it is done, then we have to wash it, and if any space is remaining when we add the antibody, then that antibody can bind to this plate. So what we have to do, we have to block the surface. So how we can block the surface? By using a non-reactive protein which will bind to the open spaces in the surface of the micro titer plate, so that no antibody will bind to it. Okay. Or if we have added antibody, then no antigen will bind to it. Okay. So antigen or antibody has the potential to bind directly with this plate. So to avoid that, to prevent that, we have to block it. We call it as surface blocking, and we use non-reactive proteins. For example, bovine serum albumin, oat albumin, aproteinin, non-fat dry milk. All these are surface blocking agents we have to use. So the ELISA can be of 2 types, qualitative and quantitative. That means from the change in color, because we are using here one enzyme which is conjugated to the antibody, and if that conjugated antibody reacts with the antigen, then when we add the substrate will be changed to a colored product, and from the color we can say the antigen or antibody is present or absent in the sample. So qualitative means only presence or absence of the antigen or antibody. That is qualitative ELISA. And from that color, the intensity of the color or the optical density of the color, optical density or intensity of the color, we can measure the quantity of the antigen or antibody, intensity of the color. So we can have a standard curve with known concentrations on the x-axis, optical density on the y-axis. So we can have a standard curve, and from this standard curve we can extrapolate the unknown concentration of the antigen or antibody by knowing the optical density. So we will measure the optical density, then we will extrapolate it, and from this we can say this is the concentration of the antigen or antibody which is detected. So in this way ELISA can be quantitative, qualitative as well as quantitative. So we can use a variety of samples. We can use urine, blood, saliva, and we can use different antigens or proteins. So proteins of virus, proteins of bacterial source, proteins of fungal source. We can use hormones, metabolites, drugs. We can use, and we can detect their presence or absence. We can also detect their concentration, both qualitative and quantitative. So what is the advantages of ELISA? It is a simple and easy technique to perform. It is sensitive. It can detect at picomolar to nanomolar range, 10 to the power minus 12 picomolar to 10 to the power minus 9 nanomolar range. So it is sensitive, and unlike your radio immunoassay, there is no risk of radiation hazard, and it can be used in a variety of samples, as we say urine, blood, saliva, etc. So these are advantages. We can use ELISA with different applications. We can detect drug abuse for drugs like amphetamine, cocaine, then benzoyl, agonine, etc. We can use them for assay of hormones like insulin, follicle stimulating hormone, luteinizing hormone, prolactin, testosterone, all these hormones, human chorionic gonadotropin, and this we use in the pregnancy test just by taking a drop of urine. We can say whether a female is pregnant or not, that we can detect. Then we can use it for diagnosis purpose, diagnosis of protozoal disease like malaria, bacterial disease like tuberculosis, Lyme disease, syphilis, brucellosis, fungal diseases like candida, and number of viral diseases we can diagnose by ELISA. The most commonly used virus is HIV. The antibody in response to the HIV can be detected by this ELISA, the indirect ELISA technique. We can also diagnose human T-cell leukemia lymphoma virus, HTLV, adenovirus, cytomegalovirus, hepatitis B, A, C, all these. So we can also detect jaundice, rotavirus, Zika virus, Ebola virus. All these we can diagnose through ELISA. We can use them in the laboratory, in the clinical research for experimental virology. We can detect and estimate the level of tumor markers, for example, prostate specific antigen, carcinoembryonic antigen, CEA, PSA. We can use them for screening donated blood for possible viral contamination. Whether a vaccine is taken or not, that can also we can detect. We can use them for forensic toxicology, food allergy. So there are a number of applications of ELISA. So thanks for watching this video. In the next video, we'll study the different types of ELISA in detail. So please like, share and subscribe. Thank you.